Twin Peaks. You know it's time to get the lab coat on. Twin Peaks. Yeah. Cause we out here being hella wrong. Twin Peaks. And you know we're never stopping till the hammer drop.
Hello, welcome back to Beak to Beak, where we make you compliant with the collectivist hive mind by informing you about the complex environment we find ourselves in. You know, Dispel, it's tough out there. It's a squid eats pigeon world, you know?
No, I'm not sure.
You gotta stay somewhat informed. Not too informed, but just informed enough.
No, I don't think squids eat pigeons. It just doesn't happen.
I mean, if one fell into the water, I would not put it past us.
Yeah, you guys are kind of like scavengers. I know.
But today we're gonna move our bodies. Move your body, girl. We're gonna move our bodies.
Shake it.
We're gonna shake it. We're gonna slime our tentacles all over the screen, and communicate to you some pretty neat stuff about bees.
What is that? What is that? What is it? Oh, no, not the bees! Not the bees! Ah! Open my eyes! My eyes! Ah!
Yippee!
They communicate to each other. They have emotions, they have feelings, they have complex internal worlds like human beings. They're alive. We need to save the bees. We can't let the bugmen kill the bees and replace them with robots. We can't let them like dehumanize our connection to our food, which is sustained by a vast dynamic system of relationships with invertebrates that are thinking and feeling. And the paper we've got today suggests that positive, effective social contagion predates vertebrates and invertebrates in terms of, I don't know, what's smaller than vertebrates and invertebrates? Like microvertebrates?
Pre-vertebrates?
Or rotifers?
Well,
or
yeah.
rotifers like waving to each other?
It's also interesting that this is like a convergent evolution. Is that? I think no, not convergent
Yeah, that was the other one
evolution.
they suggested. Yeah.
Where it's happening separately in two different areas, and it's not related to each other on the same tree, but it's happening at the same time, kind of like wings with bats and birds. What's that term?
I think it was convergent evolution.
Okay. So yeah, before we get into the paper here today, I did want to go over a brief video from Georgia Tech College of Computing that's talking about the waggle dance of the honeybee and just goes over some of the basic ways that bees communicate through physical movements and their flight patterns, different ways of buzzing their little butts.
They communicate by dancing. Quitmeyer!
Aristotle was one of the first to document the intriguing behaviour of honeybees. How is it, for instance, that a colony coordinates its work?
Okay, right off the bat, I know I say that I hate philosophy in general, and the philosophers are all weirdos, but I do really like Aristotle. He was pretty base, biology guy.
Biology? I don't know. He was sort of just like a
Aristotle is
I
the
don't remember
father
much
of
about it.
biology.
Is he?
Yes. He was
Isn't
the
he the
first
guy
one
who
to
was,
start
like,
like
Behold a chicken? Isn't he the Behold a chicken guy?
I don't think he's a
Or no,
behold
he was
chicken.
he was the victim of the Behold the chicken. I'm pretty sure.
Are you talking
Founder
about
of biology? No, he was a
Diogenes?
Christian. Diogenes is the one who pulled out the chicken, but he was talking to somebody who was suggesting that humans are featherless chickens, I guess.
Bipedal, featherless.
No. Biology was founded by Christians, actually.
No.
Because you can't have biology without genetics.
We'll have this discussion another time. But anyway, Aristotle based, love him, great Greek philosopher. The other ones, yeah, they're okay. They're not as great. French philosophers, throw them all in the fire.
Because activity,
No. Thucydides
what
is the
appears
other
to be a
guy.
random swarming mass of life, may actually... A foraging honeybee will eventually discover a new food source, such as a freshly blooming flower or artificial feeder placed by a scientist. After this visit, an interesting thing happens. Over the next few minutes, many other bees arrive at the same
They
location.
communicate to each other.
They don't travel as a
They're
group.
like, hey, psst,
Instead, each
got
bee
good stuff over
finds
here.
the food source
come
individually.
find out what I found.
Yeah, but
How
they don't
could these
fall
bees,
to the
who
death
held no
spiral of the
previous knowledge
ants.
of this site, suddenly know precisely where the
Is that what
feeder
they
was
do?
located?
No, I was just saying the bees can communicate with each other and use their superior aspect of flight to avoid the painful death that ants often find themselves in, which is a death spiral where their ant tracks circle around on each other and they just keep following that trail in a circle until everybody dies, essentially.
Is that a mental illness?
No, it's a defect in the way that they lay down their pheromones in order to track where the path from home to food is.
Okay, so it is like a defect. Yeah, I'm sure like bees probably get like similar things, like they can only fly in circles or something like that.
Yeah, I don't think it's quite as common as the ant death spiral though.
Yeah, I don't know. I've not seen
Okay.
one myself. I feel like I would be doubting myself. Like I think I would be going crazy if I saw like ants going around in a circle, following
Oh,
each
you
other.
you'd
Doing
think
the
that somebody like dosed you or you were having some kind of like epiphany, like a religious thing?
it would certainly be an experience.
Communicate amongst themselves.
You press it?
Yeah.
To answer this question, Austrian biologist Karl von Frisch devised a series of experiments in the 1940s. Researchers at Georgia Tech have reproduced von Frisch's pioneering experiments using a modern observation hive. Two feeders are placed in different directions away from the
A
hive.
highly legible hive.
At each location, visiting honeybees are marked with a small spot of paint. A separate colour of paint is used at each station, so when a bee returns to the hive, it can easily be determined which feeding site it visited. Before von Frisch, other scientists had observed that returning bees tended to waggle about excitedly in a figure-8 pattern before sharing the collected pollen and nectar with their hive mates. In this two-station experiment, von Frisch noticed that the bees returning from the same feeding source danced differently from bees that arrived from the other location. While both sets of bees perform the classic figure-8 dance, the orientation of the dances is offset between the two groups. Bees returning from one feeder perform a rotated version of the dance done by the other bees. Incredibly, the angle of rotation precisely matches the angle between the feeding stations and the hive.
Pause. Do
This
we
must
know
be-
if bees have like a magnetic organ in their head? Because like birds do. Like migratory birds do.
Mm-hmm. I don't know about bees
Do we
having
know-
a magnetic organ.
Yeah.
I don't know that much about insects, I'll be honest. I'm not a specialist.
You're not bugging out?
I'm not bugging out. I work on cats and dogs, not fucking flies and cockroaches.
Yeah, but they're feeling too.
Yeah,
All right, let's continue.
I
Maybe they
care.
do.
I care about the bees. I just don't know that much about their anatomy, if I'm being honest.
Yeah.
I know their basic insect anatomy and their arthropod anatomy and that, but I don't know like what's going on upstairs in their brain.
Or if they even have one. Or if it's connected to like their exoskeleton. I don't know.
All I know is it's not a donut, so it's not superior.
All right, press play.
Be a clue to the mystery of how the bees are able to share information about the location of food. Through further experimentation, details of the grammar of the honeybee's dance language began to emerge. The dance exploits two fundamental tools available to the honeybee. First, their ability to see ultraviolet and polarized light allows them to determine the location of the sun at all times. Ultraviolet light is able to penetrate thick clouds or fog. Also, as light from the sun passes through the atmosphere, it's polarized in a direction towards the sun when viewed from the Earth. Devices like polarized film, sunglasses, or honeybee eyes can detect this orientation and determine the position of the sun even while looking in the opposite
Oh, maybe
direction.
it is, yeah.
This
Relative
gives
to
the
sun.
bees a type of solar compass, allowing them to always know the precise position of the sun in the sky. A honeybee's entire environment seems to be constantly pointing towards the sun.
So bees are like based ancient humans in the fact that like they worship the sun god.
Um, I mean, I know that moths generally, and a lot of insects will face away from the sun, and the reason that they get caught into like human rooms or attracted to like lanterns is because they face their back to the lantern and sort of end up in a spiral coming in contact with it. So it's not that they're really attracted to light, but they're like their understanding of the world is relative to that. And so like when we've got lights out at night, yeah, it's going to mess them up a little bit. But like bees' world being oriented to the sun, like I'm not sure how that works out in like quote unquote a natural setting with like a natural hive versus what we've got in like a lab, which is what we saw earlier with like a clear lens to see like what's going on. Like they've got lights going on everywhere.
Well, bees just have a much more complex social structure that's allowed them to get to this area of social culture, where they can have a religion, i.e., the sun, where they can wrap their entire day around the sun. Whereas other insects are just they're the plebeians. They're still mucking around in the dirt. They're not finding gunpowder. They're not building culture. The bees are the ones that have the complexity enough to recognize the sun is a god.
Okay, so you think they'll be writing pretty soon?
Yes, yeah, definitely. The bees will, if we don't wipe them out completely, the bees will rise up for sure.
Bees possess a finely tuned internal clock. This clock is accurate enough for the bees to constantly estimate the new position of the sun as it travels across the sky. In this way, a honeybee can know the current orientation of the sun even after spending many hours within a dark hive. They can even take into account changes in seasons or latitudes. Inside of a dark vertically oriented beehive, the natural shared reference point is gravity, establishing both an up and a down direction. A bee's solar compass and internal clock provides another communal reference point, the sun. By pairing these two
Communal
global
reference
constants,
point,
the bees
both
form a
for
simple
morals
language.
and for direction.
Within the hive, the direction up, away from gravity, substitutes for the location of the sun. Then the angle that the bee dances compared to this up direction
Bro, they're little
is the
geniuses.
same angle a bee should fly away from the sun in order to find the target flower. So if the bee dances directly upward, other bees know that they can find flowers by flying directly towards the sun. If a bee dances 90 degrees to the left, then bees leaving the hive should fly 90 degrees to the left of the sun. A bee angling its dance towards the ground will let others know to fly directly away from the sun. As the day goes by, a bee will even use its internal clock to adjust for the movement of the sun in the sky. This lets
Let's
fellow workers
fucking
always know
go.
the correct direction
They
to
know.
travel in
That's
order
awesome.
to find food. The central waggle section of the bee's dance also contains information about the distance
The
to a
waggle
food source.
section.
Longer time spent in this part of the dance means that the food is further away. Shorter durations mean that the food is closer by. In general, a bee increases the duration of this section by 1 second for every kilometre of distance to the food.
Oh my god.
When food is within several metres of the hive, this central section of the dance will shrink,
Bro, get
causing
out of
a
my way. I'm
circular
trying to
dance.
say, like, we found these really cool flowers, like, far away. Get out of my way.
For bees, distance is actually measured by the amount of energy it takes them to travel. Thus, a strong headwind could cause a bee to dance as if the food came from a further distance away. Again,
That's
the
insane.
information contained in a honeybee's dance consists of 2 parts. 1. The orientation of the dance, which
Okay,
describes
no.
what angle to travel away from the sun. And 2. The duration of the middle part of the dance, which expresses the distance of a food source away from the hive. Other information, such as quality or abundance of food, might also be encoded within other parameters of the dance or in pheromones released by the bee. At Georgia Tech's Multi-Agent Robotics and Systems Laboratory, our goal is to work with scientists to automate the tracking and help understand the organization of multiple automata. By harnessing new computer vision techniques, we can more efficiently and effectively study the behavior of large colonies of living organisms. This information can help us uncover more secrets behind animal communication and lead to innovations in robotics.
Oh no! Fuck.
They're gonna make the robots do the bee dance to do the bee job.
So that was an interesting video. I thought it was a good, palate cleanser, refresher, whatever you want to call it, to get some information on basic bee interactions and how they communicate. And it did a lot of the heavy work for explaining some things in this article.
Lots of fake bee fans out there.
Lots of
And I
fake
don't
bee
want to catch
fans.
none of them around this channel.
Lots of fake white women on Instagram that say they love the bees, that put the Save the Bees sticker on their fucking bumper of their Kia, but then they don't plant any pollinators in their garden. They're not supporting the bees. They just want to get the signal boosting and feel the good vibes.
I emotionally support the bees because the flowers that the bees pollinate make fruit, and I love fruit.
Yeah, I support the ecosystem not collapsing mostly.
Yeah, okay.
So today we've got an article about bees, if you hadn't guessed already. The article is called Positive Affective Contagion in Bumblebees. And Dispel, would you be okay if I read this abstract real quick?
Yeah, go ahead.
All right, so affective contagion, a core component of empathy, has been widely characterized in social vertebrates, but its existence in any invertebrate is unknown. Using a cognitive bias paradigm, we demonstrate positive affective contagion in bumblebees. After being trained on colored flowers with different reinforcements, bees that interacted with a conspecific in a positive affective state were quicker and more likely than controls to land on ambiguous colored flowers, indicating the transfer of a positive judgment bias between bees. Additional observations and experiments showed that the affect could be transmitted between bees without physical contact, i.e., through visual modality alone. Our findings suggest that affective contagion may be an evolutionary widespread mechanism present in both social vertebrates and social insects.
Big news. Potentially.
Yeah, and the one thing I'm getting out of this abstract is, why am I being left out? Why are we not looking at the cephalopods and their ability to have affect contagion?
I'm pretty sure they do. Like octopi at the very least, like can signal each other
Oh
like
yeah,
affective states based on like their coloring.
you're right. The octopi are the king of the fucking cephalopods. They're always talked about. They're so interesting. Squid are just bait, right?
I mean, I don't really fish.
Oh, well, that's good.
Yeah, not my thing. We have all likely experienced moments when the joy or sadness of someone close to us quickly becomes our own emotion. This ability to match the emotional state of others, known as emotional contagion, allows individuals to share emotional experiences and information collectively, and thus is considered the most basic form of empathy and may serve as a driver for more complex forms of social behaviors. But the term emotion is often viewed as a mammal-specific concept. Here we choose to use the term affect, referring more generally to internal states with positive or negative valence, and affective contagion referring to the transfer of those states between individuals. And by valence, they just mean hedonism, because they just give these bees 60% sugar. Affective contagion has been viewed
Well,
as a
no, I
distinguishing
mean,
trait.
do you want to go over valence
Yes.
for those that don't know?
Valence is just like, yeah, excited or
It's the
not excited.
capacity of an item, emotion, or particle to interact, bond, or react with something else. In biology, the number of antigen binding sites an antibody possesses, measuring its relative combining capacity, and in chemistry, valence refers to an atom's combining capacity, the number of bonds typically formed. It is determined by its valence electrons.
Yeah, but this is affect valence.
Yeah,
This is positive or negative affect valence, and they're stimulating it with glucose, 60%
right.
glucose.
So the valence is
That's
just
a
its
that's a hit of excitement.
capacity to interact with other things. It's their emotional capacity to interact with a stimuli, is what they're saying.
No, it is a stimuli. Like, it's a reward system that they give the bees.
Right, but not all the bees are getting the reward system. So not
Yes,
all of
and
them are
they're
reacting.
yeah, so that's exactly right. The initial system, the neurophysical, like whatever, I don't know if they've got neuro, like physical, like chemical, like the same thing going on with neurotransmitters that humans do, but they've got a nervous system, they've got some internal logic telling them what's reward, what's good, what's bad. And the first bee gets that 60% glucose, goes to the second bee, somehow they're able to communicate to each other. We'll get into that later in the article. And the second bee now inherits that valence. So it's a different mechanism for understanding the world. And then I imagine when that bee would then go and look through the worlds, that they then experience that valence rewarded when they get that hit of glucose or when they get that flower. Valence isn't like potential in this circumstance. It's just like, positive or negative, happy or sad, in a very like basic primordial evolutionary sense.
Yeah,
Not in any like
we're using
chemical
it kind
sense.
of in the half biology, half psychology use of the word, not the chemistry
Uh,
use.
yes, correct.
Okay.
Evolutionary cognition sense.
And conspecifics just refers to organisms, plants, or animals that belong to the exact same biological species. So it's just members of their peer group that they're testing instead of testing it against a rat.
Yeah. So we'll get into that. Effective contagion has been viewed as a distinguishing trait of social animals such as humans, apes, dolphins, and rats. Recent evidence has also demonstrated that this process exists in parrots, ravens, and zebrafish, suggesting that the sharing of affect could be widespread across vertebrates. However, effective contagion goes beyond vertebrates. Whether effective contagion goes beyond vertebrates has been yet unknown. Examining diverse species of animals for effective contagion can reveal the evolutionary origins of empathy and lead to a better understanding of the building blocks of pro-social behaviors. Most effective contagion studies focus on negative states and infer contagion indirectly through behavioral mimicry of conspecific. Yeah. It's either conspecific or conspecific. I think it's conspecific actions.
Yeah, conspecific.
However, yeah. However, the presence of a similar behavior in both the demonstrator and the observer does not necessarily indicate that the effective state has been transferred. Consequently, to demonstrate effective contagion, a clear measure of observing an animal's affective state is required. A successful method for this purpose, widely adopted across taxa, is the judgment bias test, which is based on the idea that affective states can bias responses to ambiguous events. Non-human animals are first trained to respond differently to distinct stimuli. For example, quickly approaching one stimulus to obtain a reward and avoid another non-rewarded stimulus. Once trained, animals are provided with an event presumed to induce an affective state, and their responses to an ambiguous stimuli are then measured. Slower responses are taken as anticipation of no reward and thus indicate an induced pessimistic bias or a glass half empty bias, whereas a faster response to ambiguous stimuli are taken as anticipation of reward and therefore indicate an optimistic glass half full bias. Basically, they're saying if a bee is getting glucose, they're probably going to jump on the opportunity to guess on a flower to grab some honey from. All right, rounding out this paragraph. Only recently has this approach been applied to rats and ravens to examine effective contagion.
In recent years, bumblebees have been shown to possess impressive social cognition, including learning through observation of conspecific to roll a ball into a hole to gain a reward, pulling a string to gain access to a reward, and using social information to solve a cooperation based task. There is also evidence for affective states in invertebrates, and much of it is from social bee species, e.g., pessimism in honeybees in response to physical agitation, optimism in bumblebees in response to an unexpected sugar reward, and pessimism in bumblebees in response to agitation or simulated predator attack. Therefore, a natural question is whether social insects are capable of sharing affective states across individuals. Similar to the strategy used in rats and ravens, we combined an affective contagion paradigm with a judgment bias tasked to determine whether an induced positive affective state in the bumblebee could be transferred to a conspecific, so one of their peer members in the hive, and to explore the proximate mechanisms involved. We first trained bees individually to forage from a patch of artificial flowers in an arena. On alternate trials, bees found 8 blue or 8 green flowers, which each flower of one color contained 10 microliters of rewarding 30% sucrose solution, and each flower of the other color contained 10 microliters of unrewarding water. Color reinforced contingencies were counterbalanced across bees. See supplementary information. Within 8 alternate trials, bees quickly learned to land on the reward positive color with decreased latencies. So they were doing it quicker each time. And to avoid landing on the unrewarded negative color with increased latencies. At the end of the training, bees from all different experimental groups demonstrated similar response times to reward and unrewarded colors, as confirmed by leave one out information criterion comparisons. To investigate whether groups' differences emerged across test colors, we first performed model selection on alternative models using LOOIC, L-O-O-I-C, and applied model stacking to obtain robust model average estimates. We then computed pairwise contrast, 95% highest posterior densities intervals, for each test color. Overall, we found clear and consistent evidence of differences for the middle ambiguous colors, but not for the other colors. We thus focused our subsequent reporting on the middle ambiguous colors.
So this is the hedonism that they induced in the bees to give them this positive valence. After training, each bee underwent 5 test trials, each preceded by a brief isolation for 30 seconds in the tunnel connected to the hive and arena, during which the bee received either a 5 microliter droplet of 60% sugar water, which is the individual reward, an unexpected reward shown to produce a positive effect in bumblebees. This is the big surprise. Yippee! I got sugar water. Yippee! I'm returning to the.
I got
to
a
the hive
reward!
successful.
Thanks for the dopamine hit, Daddy!
Well, I guess it's like, yeah, give me a reward, give me like a dopamine hit, and then I'm like, I'm gonna go at these flowers like really
Hey man,
hard.
you got
That's
any
what-
more of that fucking sugar?
Yeah. Well, I mean, like they go to the flower and they probably come back. So
And they go back to the boy and they
we'll
wave
see.
their ass around. They say, "Hey, I got me some sugar. Come get some."
Yeah, that's pretty much like what they do. So they either get the 60% sugar water or nothing, which is the individual control. The bee was then released alone into the arena and presented with one flower type, or SM.
It's just telling you to seek supplementary materials to view that information.
Yeah, usually it's like FSD like 1, something like that. Supplemental figure. Okay. The bee was then released alone into the arena and presented with one flower type, either one of the 3 possible ambiguous colors varying in their resemblance to the trained positive and negative colors. So that this is like positive and negative like training, or like what's it called? Operant versus classical conditioning. There's operant conditioning.
Yeah, it's like
Colors
when they used
varying in
to
their-
shock dogs into getting them to not move and things like that, and trying to train them with shock therapy and shit like that.
That would be an added negative stimuli.
Whereas this is
Which would be a
positive
punishment.
stimuli. They had tests with both positive and negative stimuli within the same kind of timeline of when they
Well,
were doing all of
No,
that.
no, no. It's positive or not positive. Either one of the 3 possible ambiguous colors varying in their resemblance to the trained positive and negative colors. Presented in a counterbalanced sequence across trials. The trained positive color or the trained negative color. During each test trial, we recorded the time each bee took to first land on a flower after arena entry as a measure of effective state, or the level of optimism. We found that compared to with the individual control group, bees in the individual control group were quicker to land on flowers in the middle ambiguous color, indicating an optimistic bias induced by the unexpected reward.
So the way they set up this experiment is they had a bunch of different tubes. Some of them led to a reward flower with sucrose in it. Some of them led to a water flower, which did not reward them. Some of them had social controls, some of them had social rewards, meaning they were interacting with other peers. And then the last of them were just visual, meaning they cut the tunnel off so that the bees couldn't physically interact. They could only watch what the other bee was doing. And they were still able to signal their positive affect of the flower of the correct color having sucrose.
It wasn't the positive affect of the correct flower having sucrose. It was like I mean, we'll get into we'll read the paper and we'll get into it.
All right. Contagion of positive affect between briefly interacting bees. If affective contagion occurs among bumblebees, we would expect bees that interact with, observe another bee, a demonstrator, in a positive affect state induced by an unexpected reward to exhibit a similar optimistic bias. To test this, another two group of bees were trained and tested as described above, but during the brief isolation period, each observer was paired with a demonstrator bee that either had just received an unexpected reward, 5 UL of 60% sugar water, social reward, or had not received the unexpected reward, social control. Bees in the social reward group landed on flowers of the middle ambiguous color with latency similar to those with the individual reward group, but landed more quickly than bees in both the individual control groups, suggesting that positive affective contagion can occur in bumblebees. Although latency-based tasks have been effective in examining judgment bias across a wide range of species, active choice tasks can help strengthen evidence for social bias. We therefore trained two additional groups of bees in a T-maze task in which their choices were used to measure their optimism. Bees learned to find 50% sucrose solution on one side in one color was presented and found 30% sucrose solution on the other side if a different color was presented. Then prior to test with ambiguous colors, bees experienced a brief interaction with a demonstrator bee that either had just received an unexpected reward or had not received the unexpected reward. Bees in the active choice social reward group chose the high value rewards location significantly more often than bees in the active choice social control group in response to ambiguous colors, which corroborates our latency based results and further demonstrates social contagion of positive affect states in bumblebees.
Right. Okay. So I'm understanding what they're getting at with this ambiguous color. Basically, if there's like literally a mathematical midway point between two colors, and one color gives a positive reward and the other one gives a neutral reward, and like it's literally like a 50-50 whether or not it's a positive or negative reward for these bees. That's what the outcome is.
Right, they're
Like how
saying
quickly are they going to
if the
jump on it?
reward flower is red and the no reward flower is blue and we put a purple flower on there, what are they going to do?
Well, it's not what are they going to do, it's how quickly are they going to jump onto the purple flower, because it is literally like 50-50 between red and blue. I'm not sure if it actually is, but like you get the idea. If you have like a darker red violets, they're less likely to jump onto that one, or like they're more likely to line up with each other.
Right.
These experimental conditions. This is a measure of like enthusiasm.
Optimism?
Yes.
I think the term they use is optimism and pessimism.
Betting optimism, I guess. The figure shows the distinction between social reward, visual 1, and visual 2, where they hone in on the, I guess, like semiotic, biosemiotic mechanism that they're conferring this positive valence affect to each other. Okay, visual information alone allows for effective contagion in these bumblebees. Next section. Effective contagion requires one individual to display their current affective states by means of sensory cues available to another observing individual. To determine the contribution of vision in the observed positive affective contagion, we trained additional bees following the same protocol described above. They could have just blinded these bees, in all honesty. You know, just temporary blind them, bam, and see if they can like transfer this effective contagion. Might be a little cruel
Yeah,
though.
but I think they'd be a little disoriented and not as concerned in transferring information about where food sources are.
Bam! Thus confirming that it is in fact transferred by sight. All you have to do is see the social contagion. You don't even have to learn about it.
You just see the flash mob happening, and you automatically start dancing and join them.
The good vibes. Okay. Each bee was then isolated for 30 seconds of the tunnel with a specific, conspecific bee that had just received 5 microliters of 60% sugar water separated by transparent acrylic partitions positioned perpendicular to the tunnel's vision, blocking them off from each other. Each bee was then tested as described above in Figure 1. We found no indication of the effective contagion in this group, meaning that if they were face to face with each other, they couldn't transfer this effective contagion. However, the authors realized that this design may have limited the transmission of vital visual information. Therefore, in a modified setup, this is Figure 1C, and there's actually movie S1 for example clips. Ooh, very interesting. The transparent partition separating the bees was implemented parallel to the length of the tunnel, thereby increasing the opportunity for close proximity between bees, making it more comparable to the social group designs. Indeed, bees in the second visual group, compared with the individual control and social control groups, exhibited similar landing latencies to the individual reward. These results suggest that visual information alone, if provided with enough opportunity for close viewing, is sufficient to induce positive affective contagion in bumblebees. They just gotta look at each other. Take a look at that rump. That's all they need.
Shake that ass, girl. Show me the way to them flowers, girl. I want that sugar water.
Although Figure 2B is like awful. I hate looking at this. It's just not organized well.
Yeah, no, that's AIDSy.
It's organized hostile like.
It doesn't want me to understand it, that's for sure. I much more like the colorful graph on the left. I'm a big color
Individual
guy.
reward social control. Yeah, the Figure 2A is great because they actually color code the divergent behaviors that the experiment is looking to hone in on when the difference between like the flowers color, the primed color for the flowers is like 50-50 between the positive and the neutral valence.
Yeah, so to break this graph down a little bit, they broke each of the different experimental groups based on how they were able to interact with other bees and based on what reward they got. They color coded each of those with different colors and different line figures. And then they plotted that out based on the latency, how long it took them during each experiment to get back to the reward. And it looks like, as you would expect, the individual control group kept taking the longest.
Yeah. Because they were the ones just going out getting the water. But
Nothing.
when it comes to the social valence, actually, if you take a look at the visual 2 group, which is the ones that were allowed to go parallel to one another, you can see that it pretty much consistently beats every other metric or matches every other metric. So it beats visual 1 where they're face to face, and visual 1 is pretty much identical to if the bee just went out without ever seeing a bee.
Which
Just
is
like, and
interesting.
even when it's like not 50-50 colors, but actually like even when it's like pretty firmly like, okay, we're not gonna get like positive like read on this color. We've got more like enthusiasm to jump onto the flower just because they had been next to another bee, which is really interesting.
Yeah, not even like face to face, not able to touch them, in separate tubes parallel to each other, only able to look at each other, and that was the one that created the best latency times.
Yeah.
It's interesting that it's not the ones that were like face to face and tactile with each other.
Oh, the ones that were tactile with what, they just put them in the dark? That's a much better way of
Yeah.
doing it than blinding them.
Yeah, so they definitely have to use vision. Well, I mean, the tactile still figured it out well enough. You can see they're not too much further behind the visual 2. But
Well, it's not that they're figuring it out. It's like their enthusiasm to jump onto the flowers.
right, but they're getting more enthusiastic because they think that they're more likely to get a reward at this particular flower.
Yes, but not in the tactile group.
What do you mean? The
Or rather,
tactile group is transferring information about where these flowers are, not via visual representations, but by touching each other. So there is a way for them to transfer information without having to see, is what they figured out.
so as far as I understand their experimental design, the tactile experimental group is one where they deprive them. This is actually the next paragraph, if you want to take that.
Okay. Tactile and olfactory information alone do not allow for effective contagion in bumblebees. To determine the contribution of tactile and olfactory cues to the observed positive affect contagion, we trained an additional group of bees following the same protocol as above. And just for those who don't know, the olfactory would be like the sense of smell, like your pheromones, that type of thing. And then tactile would be them touching. So those cues that they're getting from the other bee aren't as good as the visual cues, even if they are able to use them to some extent. In this tactile group, each bee was isolated in the dark for 30 seconds in a tunnel with a conspecific that had just received 5 microliters of 60% sugar water. The latencies to land on the middle ambiguous flower in this group did not differ from those in the individual control group or social control group. However, they were slower to land compared with both the individual reward group and social reward group, suggesting that the tactile and olfactory information alone are not enough to induce positive affective contagion in bumblebees. So the latency then, it seems to be pretty good, but they're not actually going to the correct flower. They're going to the ambiguous flower.
Well, as far as I understand their experimental setup, they've got a variety of flowers available to the second, or rather the first, um, like bee, and it's, I guess it would be 60% for, no, it wouldn't be 60% for, so it would be 60% to prime the positive, and then the neutral would be, I guess, like a gradient of what they're measuring is the bee's enthusiasm. So the first bee got a hit of reward, a hedonistic hit of reward,
Mm-hmm.
got all jazzed up and excited, and then came back to the other bee to communicate
Yeah.
and that it was jazzed up and excited. And the
Yep.
bee required visual information. Olfactory and tactile information was not enough to convey this excited state, and so consistently they were less excited to jump on to their experimental flower to figure out if it had the sugar or not. They're transferring how they're feeling, and that's being reflected in the second bee when the two bees are next to one another and can see each other. Touch is not enough. That's why we see that the latency for the tactile condition is identical to the social control and individual control, as if they had never met a bee. It makes me feel more empathetic to bees and spiders that I crush, and I feel actually no empathy for mosquitoes.
Oh, okay. I mean, sure. Where do you draw the line at species of like where you will stop killing and like devouring them heinously, and where you're like, okay, these ones are actually worth something?
Nuisance?
How much of a nuisance they are to you?
Yeah.
Okay, I mean, sure. I can actually agree with that. I'm not like such a soy cuck libtard that I want to save all the fucking mosquitoes and all the goddamn ants and all the cockroaches in the world. But they
I don't know. Why
do.
do we even keep mosquitoes around? Like, what are they, just like bat food or something?
I mean, you know, they say that every species provides some role, and if you took them out of their environment, it would alter the ecosystem radically. But, like, can we just fucking eradicate mosquitoes? We eradicated so many different viruses and diseases. Can we aim our fucking species power at getting rid of mosquitoes? Because really, they're just a fucking plague spreader, and just a pain in the ass. Anytime we want to have a nice camping weekend out in the woods like a normal American family, they are just there oppressing us.
Yeah, I don't know. I actually don't even care about like the frogs, I guess, who would eat like the mosquito babies or something.
Yeah, but you release a bunch of frogs to eat the mosquito babies, and now you've got a fucking frog issue. Look at what happened in Australia.
Yeah, but like, yeah, I think I would probably prefer like a frog issue to a like a mosquito issue.
Then we could go frog hunting on the weekends because there'd be so many of them and they'd be so invasive that we wouldn't feel bad about it.
I mean, yeah, it wouldn't really be much of like a hunt or like sport. You'd just be like walking and accidentally like step on one.
Well, I am glad that we figured out how bees are able to transfer their emotional state to their peers. That was very
Well,
interesting.
the authors intentionally do not say emotional state. They say
I
positive
know
affect
because
valence.
they don't want to put emotions on bees. Okay, I'm doing that. I am
Yeah.
anthropomorphizing the little bee babies and giving them big, happy, and sad time emotions. Okay, I get it. I did have one final thing I wanted to talk about before we leave. Decline in bumblebee population has been pretty bad over the last 2 decades, and according to Utah State University, some reasons that it is going down so bad is habitat loss, improper apiary management, pesticides, climate change, pathogens and pests, competition, poor nutrition, colony collapse disorder, and promoting pollinators, or lack thereof, rather. So, if you can plant some pollinating flowers that are helpful to the bees in your yard, please do. And please be a part of the solution and not part of the problem.
Yippee! We love saving bees. We love having apiculture and underbrush and trees separating our agriculture instead of massive plots of land for a massive tractor to comb through.
Yeah, man, I would like some trees and some wildlife and less capitalism
Well,
and
let's get into
less
the problem
paving
though. Like,
over it.
I don't really live near farms. You don't really live near farms.
Yeah, I live near farms.
Human society is not like, built around farms.
That's true, but that's why we need to create vertical farming.
Yeah, we'll see how that goes. I haven't heard it's going well.
Yeah, that's okay. We just got to keep innovating. We'll see how it goes. One last thing I want to edit in there, statement of the findings. Their findings suggest that affective contagion may be an evolutionary widespread mechanism present in both social vertebrates and social insects. I thought that was from the abstract. I thought that was a really interesting thing because I want to see them test this theory on other social invertebrates and see if it applies to them and not just social insects.
Yeah. Oh, you know what we should do? I should find the lab that was talking about modifying ant caste behavior with neuropeptides.
Mm.
Everybody got them peptides now.
Thank you for joining us on Twin Bees. Wait, no, hold up. That's not right. Let
It's
me
Twin
fix that.
Beaks.
Oh my God, let me fix this. Hold on, hold on. Okay, there we go. Twin Beaks. Thank you for joining us on Twin Beaks. Um, this has been a fun episode about the bumblebees. I love little bumblebees.
I've never seen a bumblebee in my life.
Like and
They're like too
subscribe
low to the ground.
if you love bumblebees and want to save bumblebees. Put down in the comments section how you are a fan of bumblebees and would like them to continue.
Yeah, but we might be fucked anyways. Like we might save the bees and actually like desertify most of America anyways. So
Yeah, thanks for joining us. Like and subscribe. You can find me at Salty Squid Stream on YouTube. You can find Dispel over at Dispel Marketable Archive on YouTube. And yeah,
that's correct.
you're all
Alrighty,
beautiful.
bye-bye.
Bye.
Twin Peaks. You know it's time to get the lab coat on. Twin Peaks. Yeah. Cause we out here being hella wrong. Twin Peaks. And you know we're never stopping till the hammer drop.